Swayam Prabha. Digital India. Educated India.
Yes, thank you very much for that kind introduction. So as you said, so I am Hemachandra. I am an assistant professor here in the Department of Chemistry. Okay, so as he was mentioning, so I did my PhD from TIFR Mumbai, which is one of the premier most institutes in India. Okay, then I went on to MIT, which is Massachusetts Institute of Technology at Boston. Okay, Cambridge, Boston. I spent around 6 years there researching, and then I returned India in 2020. Okay, to start my independent career. So as he was mentioning, most of my research interest is in biomolecules, particularly proteins and enzymes. So what you will be listening today is also mostly proteins and enzymes. So we'll be having a one-on-one discussion and also some of the interesting topics or interesting characteristics of proteins. Okay, so we'll start about proteins. We'll try to understand what are proteins and what is the nature of proteins and why is it so important. So before even going into it, let me tell you that proteins, understanding the structure of proteins, understanding the function of proteins, understanding the folding and the aggregation of proteins have won more than 4 to 5 Nobel Prizes in the last few decades. Okay, so it is still a hot topic. So if you know, how many of you follow regularly Nobel Prizes in general? Okay, do you remember what was the Nobel Prize in 2024? So, very good. So, Nobel Prize in Chemistry was given to AlphaFold for not just AlphaFold. AlphaFold is only a part of it. So, basically, there were 3 different people who won, 3 different persons who won it, and one of them is AlphaFold. Basically, 2 of them shared it, and the other one is it's for computationally predicting the protein structures, and the other one is computationally designing new protein structures. So, this is the most recent Nobel Prize, which is a part of it was for AlphaFold. The other part is for computationally. One is for computationally predicting the structures of the protein. The other is computationally designing new proteins. Okay, so we'll just will not talk too much in detail, but I just wanted to give you that information because no matter and do you know when was the first Nobel Prize that was awarded for proteins? It was awarded in way back in 1972 or even before that. Okay. For showing that the proteins actually fold into a compact structure and the information for the folding of a protein is in its amino acid sequences. For just determining this experimentally, the first Nobel Prize was won in 1972. So since then there have been multiple Nobel Prizes that have gone to proteins, and still it is one of the highly undisputed leader in terms of understanding both biological mechanisms as well as trying to understand the material world. Okay, so when you hear the word protein, what is the first thing that strikes you? It's a functional unit, okay. Amino acids, okay. Complex folded structure made of many molecules, yes. Huh? Muscles, very good. Yeah, I was expecting that. Yes, muscles. It's a macromolecule. So I just write down. So somebody said macromolecules. Somebody said muscles. Somebody said amino acids. Complex structures or complex structures. Peptide bonds. Functional units. Hydrogen bonds. SS bonds. Okay. So when you hear the word protein, I thought if you are a layman, right, the most common thing that you hear about a protein is as a source of it's one of the important component for building the muscles, right? That is what you generally hear. Like, so you all know what are the sources of proteins? Fish, red meat or meat, yes. Legumes, for vegetarians, legumes, yes. milk, dairy product. Okay, I'm not very happy with dairy product, but still, I mean, the most important is egg, right? So in egg, which one is the protein part? Huh? Yes. So the outer whiter part is the albumin part is the protein part, and the yolk is the fat part, right? So basically, so you can see that proteins have a, but none of you actually mentioned. I was thinking, so when you say functional unit, what do you mean by functional unit? Who said functional units? What is a functional unit?
So it's a function of what I learned was a different type of protein can influence something. So like for example, one protein can influence whether you have curly hair or straight hair. It's a functional unit because of the different way it goes.
Okay, it's a functional unit in terms of. So it basically tells you. So have you heard of words called phenotypes, genotypes? What are phenotypes? So anything that is visible, right? Basically changes that are visible, like your change in the color of the hair, change in the color of the eyeball, and all these are phenotypes. A lot of phenotypic changes happen because of change in the proteins. Okay, so you said all this, but you didn't say there is a very important term called workhorses of the cell. So anything that you are doing currently as a biological system, right? So for example, I am able to, if I am able to talk, if I am talking and you are able to listen to it, right? So there is basically your ears are able to vibrate at a certain frequency and they are able to catch this particular sound, right? And that actually happens through a series of proteins. So there are entire series of proteins. So any biological action you take, it is nothing but it is made of proteins. So that's why they are called workhorses of the cell. For example, if your heart is beating, right? So you have your heart is beating, or you have a muscle that is kind of expanding. So you can actually kind of change the position of your muscle, or if your heart is beating, expanding, contracting, there are muscles there, right? So all the muscle movements are still controlled by your proteins. There's a protein called titin, which is the longest protein in the human, and so that kind of controls it. So although these are all important terms for understanding the protein, when we look at the protein, so when you look at the proteins in biological systems can act as hormones, they act as enzymes, right? So they are actually, they catalyze the biochemical reactions, right? They are structural entities that provide support to your skeleton and mechanical support. Then they are actually acting as your channels and pumps, and that are helping in pumping your blood. So what is a common blood related protein that you must have heard? Hemoglobin, right? So or there are other proteins that help in not just transport of the oxygen and other things, but even for transport of these ions like potassium ions or sodiums. All these ions are also transported across these channel pumps, and then most importantly, they are actually acting as your antibodies. So if you look at proteins, so functionally proteins have a huge diversity. They can act as your hormones, enzymes, structural proteins, channels and pumps, antibodies and whatnot. There are multiple things that there is nothing that a protein cannot do. That's why they're called workhorses of the cell. So if you look at a large number of diseases, right? A large number of diseases are caused because there is a malfunctioning in proteins. Right? Okay, for example, Olive will talk about it. Have you heard of a disease called Alzheimer's? What is Alzheimer's? It's an age-related disease where you have a huge, so the symptoms are forgetfulness, you will have dementia, you will have other little bit of neurodegeneration and other kind of muscular issues and all those kind of things, right? Then you do not remember your present, but you start living in past and other things, right? So there are a lot of common things that you see for Alzheimer's. And you know what causes Alzheimer's? Very good. So it's not tau protein. Tau protein is for a different disease, but this is called amyloid beta. Okay. So there is amyloid beta 42, which kind of misfolds and it doesn't. So instead of functioning, it's not like it is caused because of not functioning. It is caused because it basically misfolds and aggregates and kind of create plaques in your neurons, and because of that, your neurodegeneration happens. right? So all these are called neurodegenerative diseases. So proteins as such are not only important in maintaining a healthy cell, any changes in the proteins could also lead to a lot of diseases. We will not talk about all those diseases, but it is important to understand that they are both important for maintaining a healthy cell, and also any changes in the protein structures could lead to diseases. Yes. Protein, amyloid beta 42. Huh?
Then why is it like age related?
Because it's a protein aggregation, right? It starts, so maybe in late 40s or so it starts aggregating, but unless there is a particular size, it will not stop the neuronal circuit, right? It will not create hindrance to your normal functions. If you do a simple scanning or so, you might see small plaques which are not really causing hindrance. But with age, because the size of this increases, so after a certain age, it creates a much more hindrance than a normal thing.
Basically like resistance.
To put it in simple words, you can say it's resistance to the neuronal information that gets passed from one part to the other part. So that's why you get all these are called neurodegenerative diseases, where because you know that most of the functions in the body, except for involuntary actions like heartbeat and other things, most of them are carried out by your neurons. They're carried out by transport across your neurons, so they will get affected, and then even your memory will be lost. The dementia is one of the basic symptoms. Okay. Yes.
So, at MIT, did you conduct any research where you tried to like unfold these proteins and make them fold in the proper way again?
Of the misfolded proteins?
Yes.
Not at MIT, but at TFR we did those. When I was a PhD student there, we did a lot of those studies. So where we try to understand how to reduce this misfolding, or how to. I'll talk about misfolding also today. So basically, what I'm going to talk about today is proteins is a huge topic. I'm not talking about the functional part of it, because every protein has some millions of, there are millions of functions that proteins can do. If I start discussing it, I will not be doing justice. So I'll be talking about something that is fundamental to every protein, basically, which is called folding, misfolding, and depending on time, I'll also talk about degradation. Okay, so these are the three topics that I'll be talking about: protein, folding, misfolding, and degradation. So, as I said, so they are very, very important functionally. For example, as I said, a large number of proteins can act as signaling proteins, right? So they carry signal from one part of your body to other part of the body. They act as structural proteins. As I said, titin, a muscle. So so if you see, some parts of your body are more elastic than other parts of your body, right? And also, like for example, some parts need to be, like for example, proteins in your ears needs to be little more elastic, because they have to, the frequency of the vibration is what is going to convey your sound, right? So, and there are structural proteins also. A lot of these proteins are also structurally important. So there are structural proteins. Then other important function of proteins is enzymes, right? So you all know what are enzymes? What are enzymes?
Biological catalyst.
Enzymes are biological catalysts. You all know, for a functional body, a large number of chemical reactions have to happen, right? And these chemical reactions needs to happen with two. You can do a chemical reaction in a lab, right? But this is our lab, or basically this is a test tube, and all the chemical reactions have to happen with two conditions. What are they? They need to have very, very high specificity. That means if there is a substrate that has to be converted into a product, only that substrate has to be converted into product, not anything else. So the specificity has to be very high, because in your body there are multiple substrates at the same time. You can't really chemically differentiate them at that point of the time, right? So unlike your lab, where you're only taking a compound A and making it compound B, so you have a control on taking compound A. But in your body, there will be compound A, compound B, compound C, everything will be there, but only compound A has to be converted into compound P, right? So that means that specificity has to be there, which is done by enzymes. So enzymes are very, very specific. The other one is kinetics. The normal chemical reaction that happens in a test tube in the lab occurs very slowly. Now, if you actually compare a chemical reaction inside the cell, it is few orders of magnitude, more than a million, depending on kind of enzyme you're talking about, many orders of magnitude faster. So enzymes have to do a reaction with high specificity and also high speed. That means kinetics has to be very fast. And all the most of the enzymes, the primary building unit is a protein. There could be other kind of enzymes where glycoenzymes or something where a part of sugar is added or something, but mostly the functional part is still the protein. Okay, and then you have motor proteins. So this is something very interesting. Do you know, like, our cell is also like a city, right? Do you know, cells are not static. Cells are always dynamic. Now, if you studied something, if there is a cell that is there, something is produced here, but it has to be transported to here, right? And our cells have a lot of these tracks called actin filaments. Okay, this is like your railway track. Okay, so there are a lot of these filaments called actin filaments, which is similar to, this is like a city where cargo has to be transported from one part of the city to the other part of the city, right? Now there are these railway tracks which are called actin filaments, and these proteins, there are proteins called motor proteins, like for example myosin, kinesin. So these actually carry this cargo from one part of the cell to other part of the cell. These are just like your engines, these are just like your motors, these are called nanomotors, where they use, so basically they convert or they move, they take a cargo, and based on the polarity of the filament they carry the cargo from one part to the other part. So these act as motor proteins. So do you know what, so for any movement you need fuel, right? For any mechanical motion you need fuel. What do you think might be driving this?
ATP.
So either ATP or GTP. Okay, most of these enzymes act as, these are motors, so they are converting the chemical energy from hydrolysis of ATP or GTP, okay, and use that chemical energy to perform mechanical function. So that means they are converting chemical energy into mechanical energy, right? What are those devices called that convert one form of the energy to other form of energy or chemical energy to mechanical energy? Transformer is a general word, but in general, if you, for example, what are these elements that convert, this is what electrical energy into mechanical motion, right? The fans. What is the element that is there? Huh? Water. Motor, yeah. What is that? What is that element? What are those? Turbines. These are all machines, but there is a specific word for this. Capacitors? No, they're just storage devices. Transistor, transformers, all those are, these are called piezos. Okay, these are called piezo. Have you heard of the word piezo?
Yes.
Not pizza, piezo. Okay, so piezos are basically, they are actually converting one form of the energy to other form of energy. So all these motor proteins are actually converting chemical energy to mechanical energy and perform this, right? And the most important thing is last one, which is defensive proteins. Okay, so I'm just talking about some of the functions of the proteins. So, which is basically defensive proteins means proteins like that act as antibodies. Okay, have you heard of antibodies?
Yes.
What are antibodies? Immune cells?
If they bind to antigens such that they present on top of the compromised cells so that other cells can attack.
They are other cells. So basically there are some immune cells like T cell and other kind of cells. So where they recognize a foreign object, right? And then they bind to that so that the other cells can actually engulf them and destroy them by either by pH change or chemical reaction or other thing. But these are the initial binding elements. So there are a lot of proteins which act as antibodies. For example, IgG kind of protein. If you do any also like if you have any kind of viral disease or bacterial or some kind of thing, people you do a test which will tell you what is called CRP. I don't know if you have done that. So if you undergo any of a simple contagious disease or even a simple viral or bacterial disease, the people do what is called CRP, C-reactive protein. Okay, it will tell you how much of this protein is there. That will tell you how much of a bacterial or viral entry is there in your body. And there are these antibodies called IgG antibodies. Okay, so the essence here is to tell you that you will find proteins everywhere. So for a large section of people think proteins means something that we eat so that we can build our muscle, or it's a part of something that will help you to develop your body in a better way. But that is not the case. That's only a small part. But in principle, anything that you are doing as a biological entity is done by proteins. Okay, so that's why it's very, very important to understand in and out of proteins, because if you want to address any of the biological issues, you need to understand the proteins. So for all these reasons, proteins are heavily used as targets as well as drugs in drug discovery. So because they are controlling so many biological activities, so they are used as either drugs or drug targets. Do you know what is between a drug target and a drug?
So a drug target is something that, let's say I'm targeting something for instance, the drug just goes and limits it,
Right.
doesn't act on it. Whereas a drug will actually, release it, it'll act on that particular thing. So whereas this one will just release it.
Partly correct. Okay. Huh.
So is it like drugs are the things that actually do the action, and the drug target is the thing that the action is being done on?
So the drugs are small molecules or any molecules that actually are introduced into your system that reduce the activity of drug targets. For example, I'll give you an example. What were you all doing when COVID struck? You are in 5th class, 6th class, something like that? 7th class, okay. So you all were you were all aware of COVID, right? You're not that little that you are not aware of COVID. So if you are aware of COVID, during COVID time, anybody who is working on the drugs would have heard of something called ACE2 receptor and then spike protein. Have you heard of this term, spike protein, spike protein? Okay, so if you remember the mechanism, there were all kind of cartoons that people showed, right? So this coronavirus has a spike protein, which is S, that binds to your human cells basically through a receptor called ACE2. So this is the receptor that is there in human cells, and then this spike protein is basically a protein on the outer surface of your coronavirus, and it binds, and then it takes entry and enters into the cell. That is what you must have remembered, right? Now, this spike protein and ACE2, because they are very, very strong interactions. They are strong interactions. So now, if you can actually, if you can restrict this particular interaction, then spike protein is not going to bind to ACE2. So if you design a molecule, a small molecule that can actually inhibit this binding, that means that can reduce this binding. How will it reduce? It should have a higher affinity to the protein of humans. That means the the drug molecule that you are designing should bind to ACE2 stronger than the spike protein. Only then the spike protein will stop binding, right? So this is called competitiveness, right? So if you have your ACE2, now you introduce some molecule that is actually binding to your ACE2 very tightly, so that the new spike protein cannot come and bind to it, right? So this small molecule that you design is called drug, right? Which is basically, that is a drug. Whereas what is a target? Target is a ACE2 molecule, which is basically going and binding to it. That is called drug target. So a large number of proteins are either drugs or drug targets. Are you aware of any protein molecules that act as drugs? Drugs in the sense of medicinal value.
Insulin.
Insulin, perfect. So insulin can act. Insulin is nothing but a protein, right? And you all know, like, if you have elderly people in your house, a large number of Indians suffer from type 2 diabetes, right? So a common drug that is taken is insulin, which is nothing but a protein, right? So in this case, insulin is not a target; it is a drug that you are introducing it. Whereas in the other case, this is also a protein, but here you are not introducing the protein, but you are introducing a small molecule that will inhibit the binding of the viral particle to this. So that is called a drug target, whereas insulin is a drug. So a large number of proteins can act as, or if you can do a quick exercise. You can go to you can take a series of drugs that are available in the market, and you see most of the molecules are either drugs, either proteins, which are basically, these are called biological drugs, okay? So which are basically proteins that perform all these functions, for example, all this insulin and other kind of things, or there are small molecules that will inhibit the action of the drugs, inhibit the action of proteins, so they can act as either drugs or drug targets. But this is not just the case. So proteins have many more functions than this. So till now, whatever we have discussed is in terms of biological significance. So can you think of proteins being anything more than just biological entities?
Like non-biological
proteins are biological molecules, but the functions that we have talked about, the functions that we have discussed till now are all in biological in nature. That means they are either used as drugs, they are either used as like to improving human health or to understand human health. That means just give me a minute. So you understand the proteins either to understand biological systems in a better way. That means how they are functioning, how they are working, or you are actually understanding them to design a large number of drugs or drug targets. So all these are related to human or non-human biological systems and drug discovery. Can you think of anything that is not directly related to the human health or any health per se? Yes.
Building or architecture, because proteins are good. Building blocks, they can, if you're hurt, if the protein is there.
Okay, so they can act as a building architectures. Okay, yes. Fabrics. Okay. So you all know what is silk, right? What is silk? What is the chemical nature of silk? Silk is a pure protein. Silk is nothing but it's a pure protein. There is nothing else there, right? So silk is one of the strongest. Actually, keratin and these kind of things, all these are very strong proteins. So not just for biological or not just for health and other aspects, proteins are also used as biomaterials. Okay, a large number of proteins are also used as biomaterials. Particularly, if you see some of these proteins like spider silk, for example, spider silk is known as one of the strongest. Basically, if you look at the strength versus elasticity, right? If you calculate the Young's modulus, do you know what is Young's modulus? So you are trying to calculate the elasticity of it and compare it. This basically stress versus strain, right? So spider silk is considered to be one of the strongest material for the thickness that it has. Okay, so it is highly elastic. And then if you look at a lot of other, so this is a B-Mori fiber. This is basically a normal silk fibers. Okay, silk fibers. Then you have keratin fibers. You have collagen fibers. So all these fibers are nothing but a pure protein material which are heavily used as biomaterials. So not just understanding the human health or any health for that matter, but they also have a huge impact as biomaterials. And you know, these days a hot topic on materials is to develop what is called sustainable materials. Okay, so a large number of materials that are being used today are unsustainable. For example, you take any plastic material or any polymaterials like polyethylene, polyvinyl, PVC or anything. They are all unsustainable. What do you mean by unsustainable? So the lifetime is pretty, so you can use it and throw it away, but they are not going to be disintegrated or they are not going to absorb by the nature, right? So they are going to lie there, for example, for millions of the years. So they are unsustainable in the sense you can't keep on producing them without having an effect on the nature, because ultimately there is a limit between how much you are using and how much you are degrading. So most of these materials that we are using currently are not sustainable. And biomaterials using proteins are a new class of materials which are providing a large number of sustainable materials. Okay, so a large number of sustainable materials, basically based on the various silk fibers, various spider silk, collagen, keratin. Now all these are mechanically very stable ones. So a large number of materials are also there. So now this is all the prologue I have given you to make you understand or to make you realize. It is very important to understand what are proteins. Proteins are not just, you know, something that we eat or something that we take in just for building our muscles, but it is much more important to understand, both from health point of view, both from understanding biological systems of point of view, as well as a biomaterials, a sustainable biomaterials point of view. Now let us look at the first topic, which is understanding the folding of proteins. Okay. Now, what are proteins? Chemically, can you tell me what are proteins? Long chain amino acids. Destin? Long chain amino acids. Basically, they are. What is the chemical linkage between each of them? Peptide bond. What is the chemical nature of the peptide bond? What is the chemical bond? No, those are not peptide bonds. Peptide bond is only one kind of bond, right? Okay, so basically it is a reaction between the carboxylic acid and amine group to form a peptide bond. So what kind of reaction is this?
Condensation. Yeah.
It's a condensation or dehydrolysis. Basically, you are removing the water molecule from there, right? So a large number of carboxylic acids, basically, so what are these? These are nothing but a large number of amino acids. So what are amino acids? Amino acids have a amine group at one end and acid group at the other end, right? So you have an acid group and amine group from two different molecules that come together and form a peptide bond, right? So for understanding it in a better way, okay, let me come to that a bit. So now you have basically this is one amino acid, okay, which has COOH, and then there is NH2, which is an amine bond. So these two condense, it will dehydrolysis, and this will form a CONH bond, which is called peptide bond. And a large number of peptide bonds, when they are linked together, that is called protein. Okay. Now in a human body or in a living system, how are proteins formed? How do you think are proteins formed? You think there is what kind of chemical reaction results in a protein formation?
Trans is called translation, where mRNA bits
Right.
the ribosomes, and the ribosomes like translate them onto amino acids, which form chain.
Oh, good. So basically, most of the protein formation is predetermined. Okay. So even before you are born, right? Much before you are born, the kind of proteins that your body is going to have is predetermined. Except for, so no matter what you do, for some proteins you cannot, for some organism you cannot change the protein composition. That is because, okay. So this is called central dogma of molecular biology, where if you want to get a protein, all the information that is necessary for your protein is embedded in what is called as DNA, right? So a DNA transcribes into RNA that gets translated into protein. And do you know what is the chemical structure of DNA? The structure is double helical, but how many bases contribute to the formation of one amino acid? 4.
Three base. No, three base pairs correspond to one.
So a single amino acid is contributed by, so these are called codons. Codons means 3 bases. So in principle, your DNA or RNA has basically 4 bases, right? ATCG, or you will have AUCG, right? This is for your DNA, this is for your RNA. So they will have a huge permutations and combinations. So if I have a permutation and combination of ATCG, and each 3 bases codon, 3 bases will form a single amino acid. How many amino acids can I produce?
64.
64 amino acids. If you do a simple permutation and combination of, basically if you have ATCG 4 bases, of which 3 bases are going to give you 1 amino acid, you can have 64 amino acids. But in principle, how many amino acids do living systems have? Only 20 amino acids. So, AAA can also form, yes. So in principle, you can have 64 amino acids, but there are only 20 amino acids. That means multiple codons will code for a single. So for example, you have a single amino acid like glycine. Glycine can be coded by different codons. That is because depending on, so not all amino acids are equal in nature, right? Now, some of them are more essentials, or some of them are more important than the other amino acids. So in principle, the nature has designed it in such a way that more important amino acids are generally coded by a larger number of codons. Okay, so that's why you will have a large number of codons that are basically, in principle, you can have 64 codons, but you'll have only 20 amino acids. So multiple codons can code to the same single amino acids. So okay, and what is the difference between a DNA and RNA? RNA is okay.
As the 2' and the other side after right
Okay, why is DNA double-stranded and RNA a single-stranded?
steric hindrance caused by the extra OH group at the 2' and the RNA doesn't allow it.
So there is something called steric hindrance. What is steric hindrance?
It's like close to
Why will it happen? Like nuclei. So electron clouds, right? You have electron clouds. The electrons are negatively charged, right? So when you have two negative entities coming together, they'll start repelling each other. So the interaction will not go to the nuclei, only the electrons start repelling each other. So if you look at the structure of your DNA, this is called ATGC, right? So you have, there are three major entities, which is a sugar, there is a base, there is a phosphodiester bond. So these are all phosphodiester bond, a sugar and a base. Now if you look at all these double helix of your DNA, it is stabilized by the hydrogen bonding. So all the bases form the hydrogen bonding. Whereas if you see the RNA, I don't know if you can see this, this is the major difference. You see, this particular, there is no OH here. That's why this is called deoxyribose. Whereas here there is a OH group. That's why this is called just a ribose. Ribose is the name of the sugar, right? So ribose is the name of the sugar, which is a 5-membered sugar. So this is basically a pentose, right? So it's a 5-membered sugar, 5-membered ring. Okay, OH is the one. So when these OH, two OHs come together, they'll start repelling, and so double-stranded structure is no longer stable. It will fall apart and form a single-stranded structure. Whereas in DNA, because there is no OH at the end, so they can come close together. Once they come close together, there's the hydrogen bonding between either one of the hydrogen bond donor and the other hydrogen bond acceptor to form this kind of nice paired interactions between GC and AT. Yes.
So, I'm not so sure this is correct, but one thing my teacher said was, for DNA, it doesn't mutate beyond like a certain degree, but then RNA can mutate really fast and it can mutate to various degrees. So that's why protein could mutate extremely easily, but then generally SARS or something.
So it's partly correct in the sense like any double helical structures, you for mutations, so basically mutations, the mechanism of mutation is there are multiple mechanisms, right? One is when you're actually doing the synthesis of this can have a mutation, or you can have a mutation where you first unwind it and start mutating. So when you have a DNA, because you have a very stable bound structure, it is not easy to unwind it and mutate it, whereas RNA single-stranded, you can do it, but that doesn't mean that DNA doesn't undergo mutation. DNA undergoes a lot of, for example, a lot of cancers that you see are because of, for example, people say, don't expose yourself too much to UV radiation, right? You might get a cancer. That's what you must have heard it. Why does it happen? UV radiation is what? UV radiation is a very powerful low wavelength, high energy radiation, right? So that can actually mutate a DNA itself. So that can mutate a DNA itself, and once a DNA mutation happens, so what will that result in? Basically, once a mutation happens, that will produce a different protein sequence altogether, and that will result in kind of cancers and other kind of things. This is all in general, but yeah, it's partly correct in the sense because of the stability issue. DNA is little harder than RNA, but the huge DNA can also undergo a lot of mutations.
Yeah. Also, we say that these viruses that mutate very quickly don't have like mechanisms to check for mutations that are as robust as the ones we have.
Yes, yes. So the cellular mechanisms of viruses, it's actually kind of a both defense mechanism. So nobody actually knows whether the mutations in the virus are done purposefully or it is a random mutations, because virus, I don't know how many of you know, it is still a kind of not really contradictory, but some people don't agree that viruses are living organisms, right? So because they are kind of parasitic, they need the entire mechanism or entire cellular machinery of some other organisms to survive. So in principle, they are not living. But then, how can they mutate themselves? Are they having brains that, okay, I'll mutate it to evade any kind of new response, or should I mutate it somewhere so that a new drug doesn't bind? So it is not very, very clear, although there have been studies on this, whether it is a random mutations or is it a purposeful mutation. Bacteria, for example, you see, a large number of bacteria, I guess you must have heard of a huge problem called antibiotic resistance, right? So what is this? So antibiotic resistance means the same drug that was killing a bacteria a few decades back, which was disrupting its cells or something, is not able to do it now. Why is it not able to do it now? Because the bacteria has developed or changed its protein structures or its cellular structures in such a way that that particular drug is no longer able to penetrate through it, or the drug is no longer able to rupture it. That means it has changed the sequence by knowing that this is a particular thing that is kind of hindering my growth. So let me develop something which can evade this particular drug, right? So that is called antibacterial resistance, where a large number of bacteria have developed. So it's a huge issue. If you talk to any doctor, especially somebody who is dealing with bacterial infections, you will say that many of the or there is a prediction that probably in 50 to 100 years from now, humans might just die because of simple infections, simple bacterial infections, because that is what used to happen some 200 years back, right? Only after development of penicillin and other kind of things, people have started evading the bacterial infections. But now we are going back to the same state again, where a simple bacterial infection can actually kill you because your drug is no longer able to destroy the bacteria, because they are more powerful than any of the large diseases like cardiovascular diseases or cancers and those kind of things. But then there is that's a different history altogether that we can discuss at a later time. But I wanted to give you a bit of understanding of, so you all know how structure of nucleic acids work, right? Now the codons, so this is called as codon table, where the three, for example, U. So as I said, first DNA is transcribed into RNA and RNA gets translated into protein. So you could take three as a set and then form a codon, and each codon can code for a separate amino acid. So in principle, you can see that there are a lot of amino acids. For example, amino acid like alanine is coded by four different sets of codons, right? And stop codon. Stop codon means, so for any protein synthesis, you will have a start codon and you will have a stop codon. So in between, you have all these codons that will code for a certain amino acid. For example, now if you have, it is starting from this AUG, right? This is called start codon. The protein synthesis will, once a ribosome recognizes AUG, it will start synthesizing the protein. So once you see AUG, it will start that, and then it will start adding amino acids. For example, CCU. What does CCU corresponds to? CCU. Here it is CCU. It corresponds to PRO. PRO means what amino acid is it? Do you know 20 amino acids and their letters? You are not aware of amino acids. Okay. So PRO is nothing but proline. Okay. So CCU is proline. So similarly, again, it goes to the next codon, which is GAG. So GAG means, so it will go to the code and it will see, it's like, you look at your code and then it will form GLU. So basically E glutamic acid, then proline. So it keeps on adding amino acid after amino acid, it will keep on adding amino acid after amino acid, and finally it will form until it reaches one of these codons, either UAA, UAG or UGA. These are called stop codons. So once it encounters a stop codon, then it will stop synthesizing that particular protein. So it will start from start codon, and then it will keep on adding this peptide bond. It keep on adding peptide on, unless it sees this stop codon, which is either UAA, UAG or UGA. And then once it starts, it will stop finishing that. Then what you have is called as primary chain of amino acids. So it's called, what it just did is nothing but it has a huge chain of amino acids, right, linked through peptide bond. This is called polypeptide. So this doesn't have a structured acid. So once a ribosome recognizes a start codon, it will keep on adding these amino acids to this, and then forms and sees a stop codon, it will stop that, right? And these proteins could be very large. They can be actually as small as 40 amino acids, 50 amino acids, or they could be as large as few thousands of amino acids. So anything it can do. Okay. Now once it does that, so I'll not go into details, but I think you should know that there are 20 different amino acids with different chemical nature. Okay. So once it basically, this is what, so once it keeps on adding this particular peptide chain, this is called polypeptide chain. This is called primary structure. And the primary structure then takes on to what is called as secondary structure. So what is happening in the secondary structure transition? So you have the primary structure is easy, right? Now I have said you have a ribosome that recognizes start codon. It will keep on adding these amino acids, and then once it sees a stop codon, it will stop. So you have a chain of amino acids which is called polypeptide. All these are linked by peptide bond. This is called polypeptide. Now what do you think is a transition from a primary structure to a secondary structure? I've shown you there, right? What is the transition from primary structure to the secondary structure? So something that was a linear polypeptide. Now you know chemical bonds cannot be linear as such. So you all have heard of studied various kinds of hybridizations and other kind of things. You know Vesper. So for everything to be linear, what should be the bond angle between all the bonds? But very rarely, only like few bonds like carbon dioxide or others have this 180. All the other bonds do have a non-linear structure, right? So these bonds, you cannot make them linear because there is a lone pair repulsion, there are other kind of steric hindrances and other kind of, these are all, at the end of the day, they're all chemical moieties, right? So none of them can be linear. So once they are not linear, they need to they'll take a various bond angles there, right? So what are the various bond angles that can happen here? No. So what are the bonds that can rotate in? This is the peptide bond, right? So what are the bonds that can rotate here? CNO. Okay. So this is called C alpha. Okay. Now this bond, there is one. I'm taking example of only one peptide bond. Once from here to here. Okay. I'm only taking from C double bond O to C double bond O. That is one bond. Okay. So there is one bond that it can rotate, C double bond O to NH. This is one bond. NH to this carbon. There is one more carbon which is attached to R. So this is called C alpha. There is this second bond and C alpha to C double bond O. There are three bonds that are there that can rotate. In a one peptide bond, there is C double bond O to NH. So if I have to write this, C double bond O, right? NH, then C alpha to C double bond O, and then it keeps on continuing. This is one peptide bond. So basically, one unit is C double bond O, NH, C double bond O, R. So how many bonds can be rotated? This can be rotated. This can be rotated. This can be rotated. Now again, the same thing will continue there, right? So this is one. Basically, it can have three different rotations. But this bond is always fixed at 180 degrees. This C-NH bond is always fixed at 180. Can anybody think of what might be the reason for this? Have you heard of resonance? What is resonance? Delocalization of electrons, right? So if you actually see this, right? So there is always a delocalization of electrons between this C double bond O NH group. So for the delocalization to happen, this molecule has to be in which shape? It has to be in a planar shape. You all have studied some aromatic molecules, benzene and other things, right? So it needs to have a planar shape. It has to be in the same plane, which means the angle has to be 180. So this bond angle is always fixed. So this and this can vary. So these two are the bond angles that can vary. So just by variation of these two angles, you can have a large number of structures. And so the first point of this variation is called as secondary structure, which is alpha helix or beta sheet. That means depending on how these angles are between this bond and this bond, you can have the orientation of the peptide bond in various angles, and that will lead to what is called secondary structures, which is basically the orientation of backbone of a polypeptide. So that will lead to what is called alpha helix and beta sheet, and then it gives rise to what is called as, so this is a secondary structure and this is called, what is tertiary structure?
The three-dimensional folding causes S.
So here in the secondary structure, you only have variations of these two bonds, right? This NHC bond and CC bond. These are the only, this is called C alpha. Okay, this is called C alpha. So NHC alpha and C alpha C, these two are going to vary, and it will give, so this is called backbone. So I'm not writing about this R at all because R is the side chain. Do you know what is the side chain in a amino acid?
It's the function group that
It doesn't determine the function or the chemical properties.
no. For every amino acid, our C double bond O, our carboxylic group and amine group are the same. It's just that the R group differs from amino
Right.
acid.
So if you take an amino acid, right? So what is the general formula of amino acid? The general formula, this is one amino acid, right? NH2CHCOOH, right? NH2CHCOOH. You see only this R1s and R2s are the ones that are changing. Now, just by changing this R, for example, you take this. Okay, yeah, I'll take a simplest example of glycine. Let me see where it is. Okay, okay, maybe this because it is simple. They didn't give it. Okay, so you have C double bond OH, NH2, right? So this is C double bond OH, which is this. This is NH2, and to this carbon there is one. What is this group called? Methyl group. So if this is simple methyl group, that is called alanine. Now what is this? You have isopropyl group, right? So an isopropyl group, it's called valine. Like that, just by changing this particular R, you will have a huge variation in the amino acid sequence. That is called a side chain. Now, none of these side chains can actually help you, do not dictate the secondary structure. The secondary structure is only determined by, because I'm not considering what this R is at all, right? It is independent of this R, only this NHC is going to change, and that will give you my secondary structure. Now if I start having the information of this R, then it will form this secondary interactions will further form in what is called as tertiary structure. So this side chains will bring together various secondary structures, and that forms what is called tertiary structure, and this tertiary structures can form what are called functional proteins or further quaternary structures. Now, just imagine for a given amino acid sequence, do you think it will always, if you take a simple polymer, if you take an example of simple telephone wire, you kind of unfold it and leave it. It's also a polymer, right? It's a huge polymer. You unfold it and leave it. Will it always go back to the same structure? No. So every time it goes back to a different structure. But proteins are unique in the sense, when you have a protein and when you unfold it, so basically you understood the structure, right? Basically there is a long amino acid sequence which is a primary structure, then it goes to secondary structure, then it goes to tertiary structure. If you unfold this will again go back to this primary structure, and if you leave this, it will again come back to this tertiary structure. So no matter how many times you unfold a protein and leave it, it will always fold back to a tertiary structure, and that tertiary structure is the functional form of the protein. So that is the uniqueness of the proteins, that the structure of the protein is always predetermined by the amino acid sequence. So depending on what kind of amino acids it has, no matter what you do it to the protein, and once you release the external force, whatever you are doing, it will again fold back to the same structure. And this was first understood by a person called Christian Anfinsen, and that's why he got a Nobel Prize in 1972 for discovering this.
Swayam Prabha. Digital India. Educated India.